everyone, welcome back to DCC, the world of biology. Welcome back to all. In today's class, we are going to do fast revision of biotechnology. Actually, I wanted to take live session for this, but यहाँ पर अभी इलेक्ट्रिसिटी का बहुत प्रॉब्लम हो रही है, तो मैं प्री रिकॉर्डेड सेशन ही ऐड कर रही हूँ. So let us talk about biotechnology. As earlier also so many times we have discussed that biotechnology is a combination of biology and technology. The techniques here integrate usage of organism, either whole organism or some of the part of organisms like their cells, the enzymes or some of the biomolecules taken from that microorganisms to make the products which will be useful for mankind. So in short, biotechnology is a combination of biology and some technology. And the techniques here are used for the integration of either whole organisms or some of the part of taken from that organism. So EFB, European Federation of Biotechnology, gives definition of biotechnology in these words only, that integration of biology or use of organisms to make the useful products for mankind. And here the useful products when we make, so definitely we would be if we are integrating techniques, technology, so there are involvement of some instrument also in vitro techniques. In vitro techniques means this thing we have learned in reproductive health chapter, that when a couple are facing some problems for implantation, at that time we are applying some ARTs. So this thing we people are doing inside of lab, and then after we are implanting the zygote in the womb. The same way when we perform some practicals inside of biological lab, it is called in situ or in vivo. But when we are doing in the field work outside of the laboratory, something when we have to apply or on the large scale on pharmaceutical companies and all, at that time it is called in vitro. So biotechnology involves all the in vitro conditions. Okay. So biotechnology is a new word, but actually these things we people have been using from the ancient time. So we have ancient biotechnology and modern biotechnology. In ancient biotechnology, we know that the curdling processes which we people have been using from the our past life, or from I should not say past life, from olden era only. At home what curding we are doing, the fermentation products and all, everything is a kind of biotechnology only. But today we are doing it on the large scale, on the pharmaceutical basis. So modern biotechnology includes the integration of techniques, means instruments, on the large scale, जबकि ancient biotechnology the domestic methodologies only. Let us talk about the steps of biotechnology. First we will talk about the principles of biotechnology. And biotechnology deals with mainly two principles. These are sterile ambience and genetic engineering. So what is this genetic engineering? So in this genetic engineering we use the genetic materials or genetic molecules, and we know that we have only two type of genetic molecules. One is DNA, another is RNA. So when any organisms are of our gene of interest, means this organism we people want to use in biotechnology, so at that time its genome we need to isolate. So the first thing here is needed is the DNA or a genome or a genetic material to be used in our desired techniques, in our desired procedures that will help us to produce number of things which would be used for the big crowd, for the population, for the mankind. And for that, sterile ambience is very much necessary to maintain. So what is this sterile ambience? It is nothing else but the maintenance of sterility. So why it is needed? To avoid contamination. See what happens, like suppose the person is facing some problems related to throat infection. So the throat infection could be because of two reasons, either viral infection or bacterial infection. So if the person is having a bacterial infection, suppose cocci growth has been happening inside of the throat, so person would be having itching in the throat and sore throat. Person would not be having desired appetite, and that is why he would be not feeling comfortable. So in this condition, if we take the antibiotics, we know that antibiotic we form from fungi, and fungi is a quite a bigger cell in comparison of bacteria, and fungi is having ability to prohibit the bacterial cell. So when we take this antibiotic, this fungi फ्रॉम विच द एंटीबायोटिक हैज़ बीन मेड अप, वो थ्रॉट में उस बैक्टीरिया की इन्फेक्शन ग्रोथ को रोक देता है. सो इट्स अ काइंड ऑफ प्रोहिबिटिंग ऑन बैक्टीरियाज. सो here the sterile ambience is needed for this thing only. If we people are using anything, so it is very much necessary that the desired gene only should be expressed. क्योंकि जब जीन एक्सप्रेस होता है तो वो अपने साथ फिनोटाइप को भी एक्सप्रेस करता है, जो चीज़ हम ऑलरेडी जीनेटिक्स में पढ़ चुके हैं कि इफ़ द जीनोटाइप इज देयर देन इट वुड बी डेफिनेटली एक्सप्रेसिंग सम स्पेसिफिक काइंड ऑफ द एक्सप्रेशन फिनोटाइप्स. तो स्टेरिलिटी अगर मेंटेन नहीं करी जाएगी तो व्हाट जीनोम वी हैव सिलेक्टेड रादर देन दिस थिंग ओनली, कंटामिनेशन व्हेन इट विल बी हैपनिंग मीन्स अनडिजायर्ड ऑर्गेनिज्म व्हेन दे विल स्टार्ट टू ग्रो, सो इट विल क्रिएट सम कंटामिनेशन प्रॉब्लम. तो हमें प्योर कल्चर नहीं मिलता है. दैट इज व्हाई इन बायोटेक्नोलॉजी sterile ambience is very much necessary. Okay, now let us talk about the tools of genetic engineering and steps of biotechnology or steps of genetic engineering. So the first step in genetic engineering we need to do is the isolation of our desired DNA. We need to isolate the DNA. So as we have written here that the genetic material that we are here using, it could be either DNA or RNA of our gene of interest or organism. मतलब उस suppose generally what we use is the bacteria only, and we know that the bacteria is having its own DNA and with it extra chromosomal material that is called plasmid. The plasmid which is used as a vector. So what happens, suppose I am working on an experiment in which my desire is to produce a number of copies of insulin encoding genes. तो इस बैक्टीरिया की बॉडी में से पूरा डीएनए मुझे निकालने की ज़रूरत नहीं है. सपोज ये पूरा जो उसका डीएनए है, फ्रॉम दैट ओनली दिस मच पोर्शन इज ऑफ इंसुलिन एनकोडिंग जीन. सो डेफिनेटली आई वुड बी नीडिंग ओनली this much part which is responsible for the insulin production. So this thing, selection of our desired DNA fragment, that is called isolation of DNA. Then after isolating this DNA, we would be needing the vector in which we will insert our DNA first. Then after it would be added into the competent host, क्योंकि host के अंदर हमें ये produce करना है, जिसे हम बोलते हैं इन विट्रो टेक्निक्स ना. सो दिस 3 मेन स्टेप्स आर नीडेड. फर्स्ट वी हैव टू आइसोलेट अवर डिजायर्ड DNA. सो लेट अस टॉक हाउ टू आइसोलेट द DNA फ्रॉम द ऑर्गेनिज्म. Okay, let us talk about the tools of biotechnology. So there are 3 tools of biotechnology. One is restriction enzyme, second is vectors, and third is host. So let us first talk about restriction enzymes. Restriction enzymes are called molecular seizures because they are responsible for the digestion of DNA or cutting of the fragment of DNA. So these enzymes were first isolated from E. coli, bacteriophage E. coli, and it is named like EcoR1. So the naming of restriction enzyme, like let me write everything. The restriction enzymes are called molecular seizures because they are from the nucleases class of enzyme. The nucleases enzymes especially for cutting of nucleotides, nucleic acid, and we know that we have two nucleic acids, RNA and DNA. So restriction enzymes are specially working for the splicing of DNA or RNA, means genetic material. It was first isolated from E. coli bacteriophage, which is called EcoR1. So this EcoR1, this naming of restriction enzyme indicates like this. The first letter indicates genus, like binomial nomenclature only, and second two letter that indicates species. Of that prokaryotic cell from which enzyme has been isolated. Like EcoR1, in which Eco is the genus, Escherichia genus, coli species, and R1, this is a strain 13 from which the EcoR1 enzyme was first isolated. It's a Roman order, EcoRY strain 13. It's a Roman order only. Okay. The first restriction enzyme isolated was Hinge2, and this restriction enzymes are of two types: restriction endonuclease and exonuclease. So this endonuclease and exonuclease, they have main difference in their activity because our nucleases enzymes are site specific and activity specific. As we know that the molecule of DNA is double helical structure. So what happens, how this restriction enzyme is cutting the DNA, accordingly they have named them. If the enzyme is cutting the molecule of DNA within itself only, means from a random site at any location, then it is restriction endonuclease, means it's not site specific, it would be random, cutting would be random, from anywhere it could cut. Whether the exonuclease will cut the DNA fragment from the terminal ends only. It could be either 5' or 3', but it would cut it from the end only, means it will remove the nucleotides from the fragment of DNA from the end. Okay, so this is endonuclease and exonuclease. Now in this cutting, most probably all the enzymes like Hinge2, they cut the DNA in the sticky ends, whether some of them they cut it into the blunt end. So let us talk what is the sticky ends and blunt ends in the cutting of DNA or digestion of DNA. So the splicing will lead sticky ends and blunt ends. So what is this sticky ends and blunt ends? Suppose we are having fragment of DNA 5' to 3'. So the complementary base pair will be These are complementary bases and these are palindromic sequences. Palindromic sequences and complementary bases are having difference like that. See, as we don't make as the DNA having the complementary bases A, T, G, C. So definitely C will be bonding with G, T will be bonding with A. So this complementary bases extend this whole chain, that is called the palindromic sequences. So palindromic sequence is the fragment of DNA having the complementary strands only. Okay, so suppose the restriction endonuclease enzyme is digesting this DNA in this way. After digestion, if the fragment of DNA would be like this, then this is called sticky ends. Whether if the activity is in blunt cutting, then this fragment of DNA would be this is called the blunt end. So digestion of DNA could be either sticky end or into blunt cutting. So endonuclease and exonuclease enzymes are responsible for the cutting of fragment of DNA. Endonuclease will cut it from the random site and exonuclease will cut it from the terminal end only. Now let us talk about our second vector, which is plasmid. Okay, so let us talk about the second vector. Is called the cloning vector. Second tool. First was restriction enzymes and the second is the cloning vectors. The cloning vector generally here we use, that is called the plasmid, as we talk that it's an circular extra chromosomal material, genomic material of bacteria or bacteriophage. So generally this bacteria and bacteriophages, they carry this extra chromosomal plasmid which allow replications into the host cell. When we cut the piece of our desired DNA and we introduce it into the host cell, so it will lead the replication into the host cell. Now before going on, actually I should not write it as a second vector, actually it's a third vector, because our second vector would be the ligase enzymes. Now what is this ligase? So in the as a first cloning tool we have studied restriction enzymes. They are the molecular seizures and they are responsible for the digestion of DNA fragment. This ligase enzyme will allow joining of that cut fragments of DNA. When we will be cutting the desired piece of DNA and introducing into the host cell, as we had discussed, that suppose this is the DNA and this much fragment is our desired piece of fragment. So with the help of restriction enzyme we have cut it and now we are introducing it into the our vector. So this piece of DNA must be joined with our host DNA because it need to be replicated, because here na what is our purpose? Our purpose is to produce number of copies of desired DNA. So this much a small fragment, it will not replicate itself alone, the whole DNA fragment will be replicating. So what is our need? This fragment should be introduced into the vector and vector will allow the replication of its own DNA, and along with its own DNA this fragment will also be replicating, so number of copies we will get. So this joining of fragment, this joining, for that what thing needed, that is that ligase enzyme. So ligases are having ability to form H bonds in between the complementary counterparts of the cut fragment of DNA. So ligases are responsible for formation of hydrogen bond between cut counterparts of DNA fragment. And definitely in the complementary strands only. So this is ligase enzyme. Then comes the plasmid vector, that is extra chromosomal DNA present in bacteria and bacteriophages. The plasmid could be having 1 or 2 copies of their genome, or sometimes 15 to 100 copies also are present. Generally the bacteriophages are having many copies. Any suitable host must be having main 3 features. First is the replication site, or we call it ORI site, which we have already learnt in our genetics, that DNA replication needs ORI site, from where the replication of DNA would be going on. So the features of vector: ORI site means origin of replication, from where the replication of our host DNA would be continue. Second is the selectable markers, or marker genes also we call. So the main function of the selectable marker is to selection of transformants and elimination of non-transformants. What is this transformation? The transformation is a process of introduction of vector into host bacterium, and from the host bacterium we will be inserting it into our competent host. So second is the selectable marker, and third is the cloning vector. Recognition site. So the recognition site is a site which will allow replication of our vector into the host bacterium, which will lead to production of the number of copies of our desired DNA, and then after we would be transferring it into the host DNA. So this many things are necessary for DNA recombinant technology. Now let us talk about some vectors.